I am audible and my screen is visible. So now what we would be so today in this week, I am hopeful that you all have gone through today's this week's lecture. So the thing is that what we were mostly would be focusing on the biomarkers, how to identify biomarkers, what are the different types of biomarkers, and how to found out through different high throughput arrays, among which one example and one application using different applications of NAPPA, which is one kind of an um, high throughput microarray. So we would look at how, firstly, is that what are biomarkers and how one can identify. Secondly, is that how this high density arrays can be helpful in identifying those biomarkers. And thirdly, what are the different considerations and the challenges need to be that are there along with the identification of a particular biomarker. So now moving on, what are the so what is a biomarker? So a biomarker can be defined as a biomolecule that is capable of pointing out to normal or abnormal conditions. So whenever there is a change, so as we know that whenever there is a change in the both in our disease condition or in a normal condition, so what it happens is that generally there is a change in certain expression of a genes. So what people used to believe in earlier stages is that what is one of the main reasons for having certain abnormal conditions are the one of the is the genes getting mutated or the DNA getting abruptly changed while during different phases of replication and causing aberrations that can lead to a changes in a body and the changes in the natural well-being of an organism, which leads to different changes and different diseases. So one major thing that we generally know is that the causes of the mutations leading to different genetic disorders. So that's why genetic counseling or through the sorry to the identification of the gene or the DNA or the genomic uh, to the genomic or the DNA sequences, uh, but we can find out that whatever the if we also do a genetic or the screening of the sequences, we can identify that whether a person is going to develop certain diseases or not, or it has developed a disease or not. Next is that whenever we move ahead with the central dogma with the DNA to RNA, and whenever there is a portion of the gene which is being also in the DNA, we know that it can be your copy number alterations can be there, different kinds of mutations can also be takes place. Then other thing that is the major problem is the overexpression of certain genes or the downregulation of certain genes that can happen due to different ways, if we have certain disorders in the sequences as well as if there are certain disorders in the transcription mechanism, so then we can get an overexpression or downexpression of certain genes, which leads to have a which by monitoring certain genes expression also we can identify and understand that how whether a person is going to have a disease or not, or whether uh, that can be how much the detrimental can be. So we will see that what are the things that the biomarkers are able to express to us or able to help us in understanding or identifying. But majorly the overexpression or the underexpression of genes lead to the functional less or more amount of functional molecules, which in turn leads to a disruption in the whole cascades or the whole mechanisms, which leads us to the various disastrous effects on a particular well-being of an individual. Third is the proteins. So the overexpression of those proteins or underexpression of those proteins again help disrupts a lot of the machinery or the mechanisms of our individual and of an organism. So which leads again to overall disruption in the homeostasis or the maintenance of the system of the a particular a particular individual, and that leads to various diseases. So again, monitoring certain genes levels can help us in understanding that whether this particular disease is progressing at a rapid rate or not, or whether this disease has certain deleterious effect or not, or whether if we uh, if we target certain genes and to give a drug dose against that, so whether that is being acting or not, whether that expression has been reduced or not, all this we can measure through the gene expression. Now again, through the protein expression, we can understand and identify that whether that proteins, if what certain proteins expression we are getting a huge disruption in an individual, whether to target that protein and bring that level of the protein down, or due to the malfunctioning or the due to the having a stop codon and have a truncated protein is disrupting the function or not, and to monitor those particular protein levels helps us in understanding that what whether a person is able to getting the satisfactory option or not, or whether the uh, whether those things are there or not. So this helps us in understanding through the protein biomarker. So now, as in the previous uh, week, we had given one example of an autoantibody. So the in a disease condition, for example, in the disease, so autoantibodies are something which we know that which can be referred to as. So if I just mention it also. Just, so autoantibodies are those which are the, so you know that antibodies are something that helps us in protection against a particular antigen. So antigens are nothing. So yeah, so antigens are nothing but the foreign particles. So if I say that antigens are something which is known as our foreign particles. So now, so to protect against these particular foreign particles, so in naturally in our immune system, what happens is that to protect against these foreign particles, generally antibodies are generated or developed in our body. Now these antibodies, these similar antibodies, if starts developing against, not against the foreign particles but body's own particles. So if it is getting generated against body's own particle, so if the normal or the functional proteins that are required for the, if I say you normal or functional proteins required for body's maintenance or homeostasis, so that if body by mistake starts considering these good proteins or these normal proteins as foreign antigens and antibodies rest against that, so that gives us known as the term auto antibodies. So now detecting these auto antibodies. So if I just give you the context, for example, in the autoimmune disorder. So if I tell you about the autoimmune disorder, so you know that there are different diseases like rheumatoid arthritis and myasthenia gravis, etc., where the body's immune system mistakenly undertakes or understands our own proteins as foreign particles and starts raising antibodies against it and starts attacking it. So this type of autoantibodies. So now this type of autoantibody should be very less quantity in normal individuals. It should be in very less quantity. So otherwise, our body own proteins would be attacked by these antibodies. They will be disrupted and they will be taken out, so and our own proteins will get degraded, which will lead to the disturbance in the normal functioning of our body and leads to the disease condition. So now these autoantibodies should be very much less in quantity. Now if we start monitoring these autoantibodies, so now how we can monitor these autoantibodies? So if we have, so if I just take the arrays, and if we have the protein sequences, okay, in the array, if we have protein sequences, so proteins, so these are proteins got printed on particular arrays. Now if we flow over it biospecimen or biofluids, so if we just like biofluids, so where does our antibodies are there circulated? So antibodies get circulated in serum, right? So antibodies are getting circulated, antibodies getting circulated in serum, circulated in serum. So now antibodies are getting circulated in serum. So if we just uh, pass through the serum to these proteins of the 20,000 or so proteins which are being printed on an high density array, then what will happen? If there are if there are antibodies, so these are all normal proteins that are printed, not in the diseased condition. So ideally there should not be any antibodies present against those particular proteins, because these all are normal proteins. So if I say just these are all normal proteins and these are all functional and helps in the maintenance of the function of the particular proteins, particular individual. Now if there should be no antibody against those, because these are not foreign particles, rather the antibody should be generated against the foreign particles, the foreign particles like viral proteins or other bacterial proteins or other agents, etc., etc. So these antibodies should not be present against those particular proteins. Now, if after doing the experiment you have passed the biofluid and those particular antibodies got attached to these proteins, and then after through fluorescence if you detect the signal, then you can understand that there has been certain generation of certain antibodies against a particular protein. Now if you get to understand that what is the actual level or the function of those particular auto antibodies that are being generated, and if you see that there is an alarming increase in the intensity or the fluorescence intensity of those particular autoantibodies that has been raised against in your body, so then you will get a chance to be alarmed of that the autoantibodies are increasing, your bodies might get attacked or get prone to certain diseases, and which will be caused due to the malfunctioning or the misfunctioning of that particular protein. So these give us a clear idea. So monitoring this kind of markers, this kind of antibodies, antibodies as a type of a protein. So if we measure this kind of protein, it will give us an idea that whether our body is being prone to such kind of attacks or not, certain kinds of diseases or not. Not only this, in fact, in the so in that manuscript that we have that I have shown in the previous week, that way when we see that in the cancers also, since you know that cancer is a disease due to the malfunctioning of many systems and many processes in a body, in which also lies the malfunctioning of certain regulatory proteins which regulates or controls the processes and which creates a kind of diseases or the tumors or the outgrowth of a cell. So cancers, you know that basically is nothing but the failure of the cell cycle to control the growth of the cells, which leads to a formation of the unnecessary or unwanted cells, leading to the depriving the nutrition of the original cells or the good cells, and the tumor mass is getting higher. So now what we are trying to say is that the whenever there's a disease like cancer or something, then what happens is that also the chances of getting the normal functional proteins gets deleted, or the normal functional proteins expressions gets different. So then what happens generally is that the proteins or uh, those things like the proteins and those for those particular uh, for those particular proteins, the antibodies generated against those particular uh, uh, truncated proteins or the different mutated proteins. So that also raises in our body for those against those particular proteins. So in that manuscript that I've shown in the previous week, so one can get an idea of the cancer disease as well as can monitor the progress of your particular disease. So if there is a certain increase in certain biomarkers, if you after screening your particular patient samples and you found out that for particular protein the number of biomarkers are increasing, and those particular patients are getting uh, prone to have more aggressiveness or more metastatic behavior, then definitely you can consider and you can try to find out predict those markers through those particular screening. So that screening is known as your auto kind of an auto antibody screening. As well as in the infectious diseases, it is also very much important to identify the certain protein markers. So you I guess all of you have been aware of during the COVID pandemic, the major challenge was to understand that which patients are will become aggressive and how to prioritize their hospital with the shortage, acute shortage of hospital beds and other medical equipment. So whom to prioritize. So all this could be monitored with the expression levels of the proteins. If we can identify a sets of protein markers which are getting dysregulated with the increase or decrease in the severity of the patients, so then that monitoring those particular panel of protein markers helps us to identify and understand their causes, understand their how that those can be tackled with, and those patients can be prioritized, and those patients can be looked after. So this is are some examples of a biomarker. So biomarker term is in general is in a very in a way it's a very big term with which helps, so which can be claimed only after proven and have gone a lot of approvals and clinical trials, by mentioning that monitoring that particular biomolecule, whether it's a DNA, RNA, or protein, can let us know about various disease or various abnormal conditions. Also in an other way can also be tried to find out if we are finding that how well a body is responding after a particular treatment, then also we can get to know about the particular from a particular biomarker. So now we will look into that what are the considerations when we say that a biomarker it is a biomarker or not. What are the different discovery stage, then is the validation stage, because without the validating and without the clinical trials and without going through a set of different regulatory authorities and regulations, we cannot just pick up something on the basis of one experiment and claim to be a biomarker, because this is the one which would be responsible for monitoring a particular disease. For example, prostate specific antigen is the one which has been approved for the prostate cancer. So by monitoring prostate specific antigen, one can understand that whether a person is going to have a prostate cancer or in which stage it is. So now moving on. So what are the types of biomarkers? So as I mentioned that the biomarkers can be of many types, and have also given one example, one or two examples of the autoantibodies for different cancers, autoantibodies in immune systems, and autoantibodies, and also the proteins itself as its expression level wise difference during different infectious diseases as well as different cancers or different autoimmune disorders as well. So the mic. So if I say firstly, so by the name you can understand that the biomarkers can be diagnostic. So I have already given you an example. So if I say that PSA, so prostate specific antigen, is one of the great example of a protein marker to be a diagnostic for prostate cancer. So it is a protein marker. So you can measure this protein to find out for the prognostic, find out for prostate cancer. So now the major thing is that, so this can be a diagnostic one. Or what are the different diagnostic markers if you can think of? So just think of, if you want to confirm, uh, if you just confirm different kinds of ELISA for different confirmation of dengue test, or confirmation of any flu-based test, or in the diagnostic markers, you can also think of the hemoglobin levels for different diseases like thalassemia, anemia, to understand the levels. So in this way, this can all be a diagnostic markers. So a classic example and very much useful example what I could find out was the diagnostic markers like the prostate specific antigen, which can help us in the diagnosis of a particular patient. That type a particular patient is going to have a particular disease or not, or is having a disease or not, to diagnose those markers. So one can write the definition in such a way that those markers. So if I just mention about the particular markers, so we can say that those markers helps in diagnosis of a disease. Diagnosis, so diagnose, which helps us to diagnose, or the medical two persons to diagnose. So that gives us the marker. So that is known as your diagnostic marker. Now is the next thing. So next thing is, what is that prognostic? So what is prognostic? So prognosis is something that when the disease has condition has already is there, the individual is diseased. Now what are the things, or what are the steps, or whether that person is going to be more severe, or is going to be less severe, or what is the thing? So that is known as our prognostic marker, or the prognosis, to find out what is the prognosis of the patient, or what is the further moving off of the uh, okay. So, but the major challenge or major understanding here is that, which can lead us to understand the severity markers, or the severity of the particular patient. So, as I mentioned that if we want to have or certain markers which leads to understand whether the patient is becoming aggressive or not, certain markers if you are trying to find out and see that whether a patient is going to be in a stage where like the severity for an infectious disease. So all of these can be mentioned as a prognostic marker, or which helps on these things can be come under the prognostic markers. Third is the predictive one. So these are the ones which helps us in the prediction. whether a disease will occur or not, or the certain markers that can monitoring of those markers can uh, prevent those particular diseases or not. So these are the predictive biomarkers. So again, all these markers, I would repeat again myself that these markers, identifying those markers, having a good set of controls, and then finally validating and clinical and going through different approvals and clinical trials only can lead to the identification and the proper particular this particular identification and characterization for a particular markers to be under the umbrella of biomarkers in different categories. So these are just an overview that the how the biomarkers can actually help us in what many ways. So it can help us in diagnosis, it helps us in prognosis of a particular disease, it can help in a prediction of a particular disease. Next is the disease monitoring. So we mentioned to monitor a particular disease, that whether the disease there are certain other co-infections, certain other things that are being there or not, to monitor that particular patients. Next is the pharmacodynamic response. So this is again an important thing with the drugs. We know that how many off targets things are there and what is the toxicity level. So whether that particular markers or through which the drug was designed for, the drug has been used, whether it is being used in the body or not, or whether there are any adverse reactions, or what is the response for what is the if the drug is actually working on the human being, on that particular individual or not for that particular disease. So now, while just give you one example, that in breast cancer, so if you know that there are different receptors against which, so if in breast cancer, if you see the receptors like ER, PR, heart, etc., heart2. So these are the different receptors. Okay, so now in some patients, so if you have a drug, so for example, you take a drug targeting, just imagine of a situation if you have a drug targeting ER receptor for for breast cancer patients. Okay, so drugs targeting ER receptors for breast cancer patients that you have designed or you have known. But if you are having 10 patients, okay, just imagine a situation, you have a 10 patients, 10 or 12 patients, you given the drug targeting ER to these 10 patients. So just a minute. Yeah, so if I say that you have given a drug targeting ER to 10 patients, so which is being expressed, so there are different receptors, so you are giving the drug targeting ER to 10 patients. Now only two patients responded 8 did not respond. 8 did not respond. So now the question comes that whether that particular drug is being effective or not, whether the drug is working in a working condition or not, whether this is fine or not. But the problem is that we haven't considered the other. Some patients uh, uh, are having expression of the PR receptor, someone has a HER2 receptor, or someone is showing all the three, no expression of these three markers, of these three particular markers. So then if we target the drugs with the ER receptor, then uh, not only so that it will, rather than working and for the benefit of the patient or for betterment of that individual, rather what it would happen or what it would do is that it will lead to the off target toxicity, and it would uh, increase the problems and toxicity for the patient. So now this is an excellent example of having identifying those markers to let us know that whether this drug would work or not, or which drug to use, and what can be the pharmacodynamic response. So this is one example where we know the receptors. If there is no such identification of those, like those specific or particular receptors, rather than to try to target a generic gene or a generic protein, then how to get and how to have an understanding that whether this response is being in a positive sense or is being working or not. Next is for the risk assessment or founding that what is the particular risk associated uh, uh, and the assessment of the risk, that whether uh, as I mentioned in the pandemic, to understand that which patients or which uh, the person would be actually going for the severity. So all these kind of risk assessments can be done with the help of biomarkers. So these biomarkers are something which we can term it as a biomolecule that is helping us to have an idea regarding the abnormal or normal conditions of the individual, and can be of various types, including diagnostic, prognostic, predictive, disease monitoring, pharmacodynamic assessment, risk assessment, etc. So these are the major types or the major uh, areas where biomarkers can help us. Obviously, they are, can be helping us in other ways as well. Now, the question is, what is an ideal biomarker? So, a biomarker can be, so first thing is to have a biomarker. Second thing is to have an ideal biomarker. So, ideal biomarkers should have certain, definitely. So, the not only, so till now we could understand if we just take the term of biomarker. So bio means and marker. So you know that marker is something that marks. So here marker can be told as at points or the areas, or it can be uh, as a mark or a scale to find whether, to find the well-being of an individual. So it is marking the well-being of an individual. So a scale marker can be set as a scale, and bio because it is any biomolecules that can be DNA, RNA, or protein. So any biomolecule, and the scale to find out, or the scales to find out that whether to mark the well-being of an individual, whether an individual is there or not. So this combines the term of the biomarker, so which is we are knowing. So that explanation we understood, and we took examples, be it in the cases of diagnostic, be in the cases of prognostic, these cases of diagnostic, these prognosis, with case of predictive. Also, we have seen that how what are the different things like the monitoring of the autoantibodies, monitoring of those particular receptors, particular gene expression can give us. But the condition or the things that what we are now looking at the term ideal. And to be specific, if we uh, talk of anything to be an ideal, that means, so if we just write the term ideal against anything or before anything, so it should have certain properties that makes it certain properties so it will be having certain properties that will make it then ideal one, or the best one, which always does not become very much an ideal thing, which doesn't become very much realistic or practical, but it should be that. So first thing, that what should be, what are characteristics of an ideal biomarker should be, it should be reliable. So now the term reliable, so how can you understand that whether a biomarker is reliable or not? So for example, if we know that there are two kinds of mostly mostly what we can say is that mostly it is a two types of expression systems, or two types of understandings we can have. One is in the abundance based, one is the functional based. So we are not going into the structural properties based or the interaction based studies. So for example, if we just consider our abundance based as well as your uh, particular expression with abundance with the expression based systems. So now how we can say that whether the biomarker is reliable or not? The best way or the simple way to have to understand that whether a biomarker is reliable or not, so it if it is reproducible, reproducible across cohorts, across populations, across samples. So if it is reproducible, we can say it is a reliable. So now what is the reproducible meaning? So you know that if a biomarker is coming in cohort X, cohort Y, cohort Z, population X, population Y, population Z, if that marker is constantly being found out to be increased to across all the systems. So now in the abundance based systems, we know that the major, so if you just remember that we have discussed. So for example, if I tell you about the abundance-based expression system, specifically in the case of protein biomarkers, so we will say that it is a mass spectrometry-based, MS-based protein quantification or quantitation, MS-based protein quantification. And in this MS based protein quantification, what we are finding out is that the abundances of a particular protein across the systems. Now, if we find out that this particular proteins is coming across the strategies, across the technologies, across the labs, across the population, to be very much coming as a in a reproducible manner, that it is still coming as a highly if a protein is highly upregulated and it is coming across the population, then we can say to be a kind of a reliable one, that it does not belong to a particular strategy, it does not belong to your particular uh, sample preparation, it does not belong to a particular population, and it will be can be reliably done. Next is the safe and easy to measure. So now when we talked about few different technologies, so once what we told about the autoantibody screening. So now the thing is that the biomarker is something when we are saying that it is able to help us in getting understanding that whether an individual will be prone to that disease or not, or if that individual is already in that disease condition, whether it will become an aggressive or not, then it should be definitely safe and easy to measure. That it should not be that something which we, so for example, if I say that that if you just go to a hospital for a regular clinical checkup, the doctors routinely say or administer to you to get your blood test done or urine test done, but it is very rare case that a doctor will suggest you to go for a cerebrospinal fluid test, because there needs to be a puncture needs to be done, taken out the CSF and then try to find out the markers. So now getting the markers out of the cerebrospinal fluid, and if you find out certain markers on that kind of systems which are very much invasive in nature, in the fluid or any biospecimen which is in the invasive, so that makes you to that is not very much safe as well as not very easy to measure. So the whole focus of the biomedical research or the clinical research has been shifted to a focus where one can get in a very non-invasive way, not in the invasive way. In fact, blood-based tests can also, if you can replace the blood-based test with also very simple invasive, non-invasive like techniques like saliva, or if you have sweat, or if any other such thing which for which we don't need to have a puncture, and if we want have to injured, and if we want to take that biospecimen out, that would be very much useful. So also that this puncturing and then taking out of any biospecimen poses also many risks and many threats, specifically if the hygiene conditions are not maintained well in a hospital. So all those poses a risk. So it should be safe, easy to measure. So for example, so people are now looking at various biomarkers from saliva, and also a paper a manuscript came where one could detect, where researchers have reported that uh, they could find out different markers in saliva, protein markers in saliva, with the help of MS-based mass spectrometry based proteomics, to have their changes for a breast cancer patient. So that can be taken forward, which can be taken forward as a diagnostic marker for breast tumor. So you can understand that having certain markers in saliva or in sweat, if we can detect and if we can identify, then definitely it will be very much good, and it will be very much safe, and it will be easy to measure. Second, third is the sensitive and specific. So sorry, just sensitive and specific is certain thing which I will uh, talk in detail at the end of this thing, that what is the sensitivity and specificity. But in briefly, what we can say is the sensitivity is something to have as many as have as many as the correct responses, that how many that whether by monitoring this particular marker. So if you say that you are monitoring, for example, PSA, that is post specific antigen, which is an example of having been approved as a biomarker and is being used for the diagnosis and the understanding of the prostate cancer. So now that sensitive means that the patients having the prostate cancer, as many as positives are there, should be get reflected from the monitoring of this expression of this prostate specific antigen. And the specificity is that those who are not having the prostate specific prostate cancer should not be detected as a prostate cancer by monitoring the prostate specific antigen. So it should be reliable, it should be very much safe and easy to measure, and it should be sensitive as well as specific. then it can be applied to both uh, diagnosis as well as staging prognosis. So we had said that prognosis is something which is very much crucial in a way that prognosis help us to get an idea that whether a patient is monitoring towards an aggressive ones, should the patient be very much focused upon to look for certain markers or certain diseases, whether that should be actions should be taken or not. But so if a marker which certain markers, I'll just give an example. Like for example, a marker whenever there's a transition from a, so if I say transition from, if I say just transition from normal to diseased condition, a certain marker level gets increases. So what I mean to say that you, when you are transitioning that you are in a normal condition. So for example, best thing would be the your immune cells. For example, just take an example of the cells of the basophils and all those immune cells. So you have got an infection, the number of cells gets increased, and for fighting against the particular microbes. So you can take the responses like the interferons and those particular antibodies. Now, when you have gone to the disease condition, you have a certain increase on those particular antibodies uh, and those markers. But in the if it happens so that, for example, any marker X which you can monitor when you are going to have a particular disease, or when you have a particular disease, and you can find out that you have got that particular disease, but it does not change as it progresses. So if you go from non-severe to severe once as well, but the marker level expression does not change and can is remaining the same, then what is happening is that we cannot take it. We cannot, and in a same disease, if there is another marker Y which not only changes after the disease condition, but it is also being constantly changing as the patient is progressing, whether it is progressing in an onward positive sense or a negative sense, that is whether the patient is progressing towards severity or towards betterment. It is also constantly changing and giving us an idea, giving the clinicians an idea that whether this patient is progressing or not, whether it is getting getting response, giving response to therapeutic therapies or not. Then it is getting differently, then that marker will be definitely chosen, because it gives us an idea for the diagnosis, staging wise prognosis, that whether is the patient is going towards the betterment or is going towards the disease condition. So this is the major thing. So this will give the status, one as a biomarker, one as an ideal biomarker. So it will just differentiate the two rational or the two uh, things with the very effectively, that which one is a biomarker and which can be taken forward as an ideal biomarker and should be studied to get the cohort done. Next is the cost efficient for follow-up. So if we need to monitor these particular markers repeatedly, then it should be very much cost efficient, because otherwise, so for example, you know that the having uh, understanding for the diabetes level, if monitoring of blood sugar level generally remains very much difficult, or very much cost is associated with it, one would not prefer to, or one would not be able to do that. So it should be cost efficient for follow up and modifiable with treatment. So it's the best again ideal biomarkers should be that whenever the treatment is given, if that person is responding to that treatment, then it should get its expression changes or get its value come back to normal level or towards normal rate. So this gives us a hint that whether an individual is monitoring and whether is responding to that patient or not. So the major difference between a biomarker and ideal biomarker is that a biomarker, definitely an ideal biomarker should be a biomarker with a biomolecule that is responsible to understand the various conditions of the disease, and the disease conditions can be monitored, whereas ideal biomarkers are some of having some of the more features like should be reliable, reproducible, safe, easy to measure, sensitive, specific, can be applied to diagnosis, staging, prognosis. Then we can have cost efficient. It should be cost efficient. It should not be invasive in nature. It should be as much as non-invasive in nature. The best is if we don't need to take the specimen out and can use biofluids like saliva, sweat, etc. And it is modifiable with treatment. If once the treatment is getting responded by a patient, then it is changing accordingly, and one clinician researcher can get an idea what is the change. So this is the concept of an ideal biomarker with certain more qualities. Now, if we talk about protein biomarkers, so we have already talked about the proteomics field. So proteomics field give us an excellent idea about with various technologies we can have a knowledge about the protein expression across the normal tissues, normal cohort and the diseased cohort. We can understand the PTM structures, sequences, protein-protein interactions, structural proteomics, functional proteomics, and so on. So now in the protein expression, what we know that whenever we talk about expression, so one is if we just think of the two major classes when we are talking about a protein-based biomarker or gene-based biomarker, one is and specifically for the proteins, whenever we are talking of the protein biomarker, one is the abundance-based, one is the functional-based. So if I say that if we just look at it, so whenever we are telling about the protein expression based markers, that is the abundance based markers that we have looking into. So in the mass spectrometry based proteomics, or the what we can do is that we can identify that what is the level of the particular protein or particular sequence is present, the abundance, and if we found out when we compared the disease condition with a normal condition, and if we find out that okay, whether that particular protein, and if we say that okay, particular protein is going to have very much effect on the on that, but if there's a highly upregulation of a protein present, then what we can say definitely that okay, so these particular protein sequences or these particular protein expressions are getting changed, and if we can monitor this, we can identify that whether this particular person is developing the disease or not. So this is the kind of an example of abundance based. Now, post-translation modifications and the protein-protein interactions are those which are related to the properties and the interactions between the different proteins. So, if we tell that, okay, so there are different interactions, different modifications, PTMs are present, so then that will definitely give us an idea and an opportunity for understanding this particular uh, in a much better way, that if there is a certain phosphorylation change that is associated with a particular disease, then how to stop that phosphorylation and to monitor that phosphorylation levels. Then is the in the structure and the functions of proteins, proteins normal behavior depends on quite on the structure relationship or the and the functional proteomics. So these are the major things one we can classify the abundance based. Another one is functional based, which can be taken forward as a protein biomarker. So this is what gives us an overview of the different protein biomarkers and that different proteomics technologies which we had talked about to some extent in the previous week, which we had seen that the MS-based proteomics and in the difference between the MS-based proteomics and in the gel-based proteomics, whereas in the MS-based proteomics, we will find out that there are different quantification methods like level-free quantification, level-based quantification. Accordingly, we can find out the abundance and can try to select out those particular features from a array of 20,000 around 20,000 proteins and to find out some proteins which would be useful for monitoring a particular disease. So now the major thing is that before understanding that uh, getting all these things, so now if we talk about abundance based or a function based like the protein microarrays or those things, so there is the need of the express purified proteins. But before going to that, just think of to have feed proteins this much, this much, this much. So some proteins are having basal level expression high, basal level expression high, some are having basal level expression low. Okay, so basal level means that in the normal condition only, basal level expression high, basal level expression low. Now to compare, we should understand that which particular. So if you just understand, so there are many such data is there, and we need to identify that one particular marker above the wall. So this analogy you can take. So you have room, consider it as a room with 20,000 files. So a room, you are considering a room with 20,000 files, and the file present here is that one file we are looking for. So among all those files, you would be trying to find out that particular file kept in that middle of a room. So this is how this finding in this kind of abundance based expression of those things we are trying to measure that what is among the 5,000, 6,000 or 7,000 proteins identified, what is the particular protein marker that can be used and that can be taken forward for the actual identification or actual identification of markers. So now this is, I guess, everyone of you are clear about the particular concept of finding that particular file or that particular thing from a wide set of materials that are present to get exact that particular file that will lead us to the understanding of the mechanistic use very much. And second is that to have a biomarker that can be taken forwarded for different clinical studies. So that is what remains very much challenging. Whereas to have the properties or the interactions or the to have the functional based proteins are relatively easier with knowing the particular genes for those different 20,000 proteins, which can be taken forwarded easily. So if you have those for those particular 20,000 proteins, and then what we can do is that we can easily find out through various ways the modifications and the interactions, and then what we can do is that uh, we can try to find out what is the status. So that's why we need to have a very robust thing to have to identify the expression levels of various proteins. Second is that to have a knowledge of those analyses, and to have particular to use different machine learning based approaches and other feature selection algorithms to extract out those particular proteins or markers which can be used for differentiating your disease and the normal state. So now the major things that are required in both abundance based and the expression based and different types of things. So to have an expressing the purified protein. So in the abundance based, if you think of the particular microarrays and all those interactions, so we cannot afford to have those in a very short notice, because we need to have a express of the unified proteins, purified proteins. Then we need to perform the cloning and then perform the validating sequences, and then protein expression based clone system is to be formed, which is to be protein expression based clone repositories, which would be used for expressing in the particular what you say that expression vector, which would express your particular protein. So the major challenge lies here to express the purified proteins, because if the proteins are not purified enough, then it won't show that particular reaction, and it can be a false positive. As well as to have an expression of the purified proteins, we need to also have a very robust idea about uh, to get that particular protein out, and then to try to get it in the array. So this is a challenge that needs to be associated when we are uh, having to express those particular purified proteins. And all the proteins are also not very much easy to clone, and then to have the expression system out, and then the purifications. And also if we need to do in the scale, so what can be the scale, what how to do that, because if you are trying to monitor 100 patients, so what is should be the scale, and then how much quality checks is to be ensured that whether that particular proteins that you have purified are purified and does not contain any host protein. So we need to have an idea to get host proteins association with the expressed protein. So if we want to have a association of the host proteins, which we need to have. So what are the host proteins associated with it? Then the purification. So then the cloning part, along with that validating the sequences. So as in the lecture you could see of the DNA SU. So if you just go and search DNA-AC from Arizona State University. Professor Laver from Arizona State University has got made a huge clone repository. So suggest you all to go through it in the DNSU. So where you can get the list of all the clones and the repositories and how it has been done, with what control regulations have been measured and have been taken, so that there is no such problem associated with those particular clones and can be easily taken forwarded for maintaining the expression or maintaining those things for the particular protein. So I'll just show you. So the DNSU, so it has a huge clone repositories. So it is having a huge clone and repositories, which will help you to get those particular proteins expressed out with just line in the expression vector, I guess. Then protein expression based clone repositories, which Professor Joshua Laber has taken a great initiative and has done almost for all the proteins across the different states of India. So this is sorry, which has Joshua Laber done with the different proteins sequences of the different clones and the repositories across the human proteins of the 20,000 almost 20,000 human proteins, which one could do and one could take and can be trying to find out that protein expression based, that what is that expression of that particular proteins and can be used. So if you can use those clone taken and can be used across. So this is those particular expression clones with verified sequences of those particular genes. So the major challenge or the major thing that is remaining here is that one needs to have in the clone repositories. So almost of one particular protein from one gene sequence, one CDN sequence, one particular protein from cDNA or gene sequences. So if so Professor Laver in the DNSU, what has done is that has taken and verified all those sequences and through different cloning systems has expressed those particular proteins, and that give that protein sequences there, through which one can uh, uh, take forward and can be used for different expression of different proteins and can be used for different applications. So this is one. So to express the purified proteins, then is the cloning, then the validating sequences. So if whether it is an abundance based or a function based, the major challenge in the protein is that to have the expression of the purified proteins. So these again now you can ask me that to express and purified proteins and then cloning and then validating sequences. So all these systems are in a way cell-based systems. So in the previous week only we have touched upon the two different expression systems. One is cell-based and another is the cell-free. So if we are taking is that as an cell-based expression system. So cell-based expression system is something which helps us to get the purified proteins out. Similarly, there are other expression systems as well. So here what we are saying, here we are saying that it is a cell-based expression system, and you can definitely ask me the question that should everything be we should go through these cell-based expression systems or not? Definitely not. There are cell-free expression systems of which we have talked, which we have seen in the previous week itself. That using the cell-free expression systems as well, we can easily go and identify, and we can do the experiments. So we will be talking through for that only. But before moving on to the cell-based expression systems and the cell-free expression systems, I'll do just a quick recap, like the different expression systems of the abundance based and the structural functional based and the different interactions and the properties based. Okay, so now what we are saying or what we are doing is that what after we are getting the abundance based protein expressions and the uh, uh, and having those particular expressions. So now we will do just a quick recap. So we are focusing mostly on to identifying or to have those protein biomarkers. So high throughput protein microarrays. I'll just give a quick overview or a quick recap of those protein microarrays. So we had talked about it in the previous week itself. So just to have an idea about what are the different techniques that can be used through of specifically the microarray based techniques, which would help us to get an idea regarding those particular which could get to the particular this high throughput protein microarrays to have the understanding or identification of certain protein biomarkers. So what are those particular protein biomarkers that can be used across, so that what we have uh, we are going through or we have gone through in understanding. So now we will do just a quick recap of that. So that what we had said in the previous uh, week itself. So one thing is that the direct labeling. So through the to have the high throughput protein microarrays, so we should get to that different ways. So one is the direct labeling. Hopefully there are no questions till now. So that helps us where it is the antibodies are printed on the glass slide and the proteins are directly labeled with the dyes measuring the fluorescence intensity. So antibodies are generated and antibodies are printed on the slides. So now you can understand that what are the challenges that can remain associated, of which we have also spoken of in the previous week. So the direct labeling that is happening is that antibodies are getting printed, but still this particular division would help us to get on the same page of what we are talking about. So if you just don't recall of the cell free and the cell based expression systems, so because we have told that we need to have those biomarkers, and protein microarrays are one of the most technique high throughput technique which help us to so in the one is the mass spectrometry based proteomics, where one can get a very high throughput response and the expression levels across samples. Similarly, in the microarrays, that's the one another techniques which help us to get that. So now the question that remains pertinent is that expression systems are remain so close problematic, and what are the ways with to have the sequences and then the considerations, etc. So now we are moving towards the high throughput protein microarrays. Now again, protein microarrays, what are the different types and what are the different challenges? So first thing was that of the direct labeling, where the amino acid, where the antibodies remain, uh, where the antibodies remain very much printed on the slides, and the proteins are labeled with dyes measuring your fluorescence intensity. Next one is the sandwich immunoassay, where you have the antibodies, then you have the protein, and then you are measuring it through the secondary antibody attached with the substrate. So that is same as you do with the sandwich ELISA. And the reverse phase protein array, which we had already looked upon, is that the cell lysates are printed on the glass slide and the fluorescence is captured through a primary and the secondary antibodies with the which secondary antibodies are as substrate. So here the cell lysates. What are the cell lysates? So cell lysate, if I just tell you that, so cell lysate is nothing but which contains your proteins of interest. Proteins of interest, and it is being measured through antibodies. Proteins of interest, and it is measured through your antibodies. Now another is the peptide arrays or the peptide fusion tags. So I'm not going into the details because in the previous week only you can find out, but I just wanted to recap it so that we are on the same page in understanding this other technologies which we will be looking on. So in this today, one thing that we looked upon is the biomarker. The next thing that we are moving towards to the is the type of microarray technique that have been developed at by Professor Joshua Leber, and he helped in getting the uh, designing the steps for these cell-free expression-based systems on the NAPA, or the nucleic acid programmable protein array, which minimizes the problems or the challenges associated like with having a great purification system, great expression system, and then finally getting the cloning and then doing the verification and the validation steps and getting the extract purifications. Then also in the clone certain mutations happen and etc., and to have the natural condition of the proteins, to maintain the shelf life of the proteins that have been generated and cloned, and after it has been purified and extracted out. So all these things can be uh, uh, can be uh, can be adjusted by with the NAPA or the NAPA arrays, and obviously multiple spotting techniques which towards some are very much being used as a technique. So one again, so not only in the full proteomics what we talk about abundance based or the function based, so in the arrays also we can get the abundance based arrays and the function based arrays, and uh, which will help us. And the high throughput, always the challenge lies with this kind of microarray techniques is that high throughput with the small amount of analyte. So this major advantage of this microarray technologies as these are very much high throughput and a small amount of analyte, but uh, it's required analyte means your biological sample, and the protein purification remains a major challenge. Okay, so hopefully there are no questions till now. So the major thing for an ideal protein microarray. So now we had looked for ideal uh, biomarker. So as I mentioned, that anything ideal means to have certain properties that makes it very much useful and is being very much used by the researchers and the scientists across. So now once to find, so you can frame this in this way. So you are having, you should find ideal biomarker, so which is so if you just give a description again: reliable, reproducible. It should be reliable, it should be reproducible, it should be uh, ideal biomarker should be reliable, it should be reproducible, it, it should be used for diagnosis to prognosis, stage wise prognosis. It should be non-invasive, so it should be measured from ways where like not from it should be avoided like CSF and all, tissue biopsy and all. Rather, it should be used like in the sweat, saliva, or serum. Should be as much as non-invasive as possible. Then it should be cost effective for having monitor those should be very much in a cost effective sense. So all these are the reasons for to have one stages of ideal biomarkers. So now we are looking for to have ideal protein microarrays to find out ideal biomarkers. So what it should be having? It should be having a high density. So high density arrays are something which is being spotted closely and are spotted in a greater amount. So it should be about the high density, having a small volume, then the multifunctional. So an ideal protein microarray should have this particular functions or should have this particular uh, quality. So now you might be asked a question. You might be facing or can be asked a question that which of the following properties, which of the following properties properties should be or should not be, or should not should or should not be there in an ideal microarray. Ideal microarrays to find ideal biomarkers. To find ideal biomarkers. So which of the following qualities should be there in an ideal protein microarray to find an ideal biomarker? Should or should not have. The question can be framed in both the ways. Either should have or should not have. So it should have high density. Then you have the small volume. Then the multifunctional. Then is the main thing is that milieu, mammalian milieu. So milieu means the conditions. So it is the meaning is conditions. Mammalian milieu means the condition is that it should have a mammalian condition to have a natural folding. So the proteins, if we are trying to find out between a antigen and antibodies reactions, though it occurs through different reactions like your van der Waals interaction and different interactions. So, but it should have better natural folding. So the major challenge lies whenever we work with the proteins is to have a proper folding of the proteins. Then only its actual functionality and its actual things can be easily monitored and understood. So to have an mammalian milieu, so milieu means a condition to mimic the mammalian condition to mimic mammalian condition to mimic mammalian condition where the proteins gets naturally folded and can be identified and can be uh, measured in a very correct way, in a very measurable way. So that's an important point. Next is the protein extraction and the purification strategies. So this is a major challenge whenever we are using a cell-based system to extract the protein, first to express the protein, then extract the protein and the purification strategies. Then what about protein stability and functionality? How much proteins can be stable and can remain functional? So this is very much important for us to understand and have in place. So the protein should be stable, protein should be functional, should be stable, should be functional, should be stable and should be functional. So ideal protein microarrays. So what is the stability and functionality of that particular protein? So in the proteins, so first things are on the basis of the properties of the arrays with having high density and arrays and to have a small volume of analytes and to have a multifunctionality. So next is the mammalian milieu or the condition to have the natural folding and the protein extraction and the purification strategies, and the protein should be stable and functional. So now to address this kind of different challenges associated with the protein expression, extraction and the purification, it was nucleic acid programmable protein arrays on NAMPA arrays that was developed by Professor Joshua Leber, as you have seen in the, should have seen in the lectures, and otherwise uh, also we have discussed. So this is a kind of a cell free expression system. So we don't need cell free cells to express. So what is that? So as I have already spoken and as I have already mentioned, so to have the DNA constructs, so the basic uh, what I can say is that basic assumption is that one particular cDNA molecule, that one particular cDNA molecule is coding for a particular gene or a particular uh, of a particular gene, that which you are getting out, that if you are getting a particular gene and if you are getting a particular uh, protein out, so uh, that one particular gene sequence or the cDNA sequence is coding for one particular protein or one particular thing. So then what we are saying or what we are taking into account is that particular DNA templates or the constructs or the clones or the repositories which has been maintained and have been designed by Professor Lever with very minute details and very much verified with the sequences. So what we are doing is that on the chip we are having the purified DNA template, we are having the BSA and all the different extracts that are required for the production of the proteins. And what we are doing is that we are keeping an anti-tag antibody. So this is a tag that is there or kept on those slides for each protein to get purified, to get transcribed and translated. So once its DNA has been, once the protein has been prepared, so that it goes and attaches to that particular place where the antibody is there. So this helps in getting or attaching the proteins to that particular antibody, as well as it gives the functionalities. So what is that functionality? So it also gives us the uniformity that the protein is getting placed across the slide, that the proteins are, so that the proteins are placed across at a particular position where it should be according to the antibodies there. So the proteins are getting attached to that particular region or place only. So that gives us an idea that how we can compare between the two particular proteins or two particular samples. So we are adding this cell extract and this it's newly synthesized protein. So cell extract are the ones which gives you the particular like the lysate of the rapid reticular lysate or those extracts, which gives us the necessary factors to translate into the protein. And the newly synthesized protein, as you can see, is getting placed into the protein tag for the antibodies that is there to capture those particular proteins, and then you will place over the query of the samples query over the slides, which will take you to the understanding of those particular expression. So this is the concept to have structurally and functionally correct protein molecules without going into the steps of requirement for to have your particular where you are needing and wanting to go unnecessary in the path of cloning, then to have an unnecessary taking out your particular uh, purification. Then, as I mentioned, that the challenges remain associated with the attachment of your host proteins, which will not give you the exact structure and function of a particular protein. Rather, it will be a different one. So, all those things can be taken forward and can be just replaced by using a very novel method of a cell-free method where you have a purified cDNA template with an assumption that cDNA is coding for a particular gene and a particular protein. We will giving the possible all those requirements for a gene and to get into the protein, and then once you have done with the newly synthesized protein, either that antibodies are remain tagged very well, proteins will get and attach so that the proteins are getting attached to that correct location and position, and then after query of the samples you are passing through it, you can try to find out the expression and the levels across your particular proteins. So what are the some of the advantages of NAPPA or the cell-free expression system, among which NAPPA over other cell-based systems? First thing is that no need to express and purify protein. Proteins are expressed in mammalian. So whenever we are doing any kind of innovation or when uh, we are trying to find out a certain particular proteins, so certain particular levels. So definitely the thing that is required here is that we need to uh, have to overcome the challenges associated with the traditional methods or the known methods, and then only what we are getting is that we should get to have an expression, and that if that challenges is overcome, then only that innovation and the those things will be of any use to us. So for example, those advantages of NAPA over other methods are to have express and purify our so there's no need to express and purify proteins. Proteins are expressed in mammalian milieu because we are either giving an yeast extract or the rabbit reticulocyte, which is giving you the equivalent mammalian milieu or the mammalian. If we pass to the rabbit reticulocyte in that to express the proteins in a cell-free manner, so that is expressed in a mammalian condition to helps to have the natural folding. Third is that it is having a consistent levels of proteins because you are just using the CDNA template and giving the necessary conditions for all the proteins to be get translated or transcribed over there. So no issue of the stability of proteins, access to any proteins with the available cDNAs. So we can access any proteins with the available cDNAs, and the expression systems with the immobilization and detection schemes. So expression systems with the immobilization detection. So if we are having the antibodies, it will get immobilized to that particular amino acid region only or the particular position, and to have the detection streams, and the correct proteins will be detected through different antibodies. So this is how overall NAPA workflow is there. So I'll just go through it and try to give you an overview of how the NAPA things works. So what we are saying that we have a T7 recombination cloning, sequencing, and high throughput DNA extraction with going of the with the particular things. Next is the site functionality, high density, and feature pricing. So this is the cloning, and then the spotting of those arrays. Now comes the second part, or the cell-free protein expression part, or the quality control with the picogreen and the protein, and the assay will be done through secondary antibody, and then is the analysis by the background subtraction and the data normalization. So you have the halotech system, multiple color tags, and then so these are some of the improvements that is happening with the nanovels to have the multiplexing and to speed spot, and the major challenge is that the spotting and those things. Then the mammalian systems which will give you the improved folding. So that's the major point that what I was trying to measure. So you are passing through the cell lysates and you are giving trying to give the mammalian systems for improved folding. Then the level free strategies and new data processing methods. But there are certain challenges as well with the high density arrays, with the chemistry, cross linking, and the purification and the printing. So, as I could see that high density feature printing, reproducibility test, size functionalization, GST tag, sequencing. So, these all are very much important, and these all are very much responsible to have a very good high density arrays which will give us a great functionality and get it will help us in experiencing a huge good quality of the data to have across the sample cohort, so which generally gives us. Then is that the quality control system. So this, whenever we try to do something and try to publish or try to find out, we should have these particular systems in place, and we should have the idea that whether our particular systems are we are getting or not, or whether we are getting that actual proteins are getting purified and getting placed over antibodies or not. So these kind of things should be there. And then finally, definitely assay developments and all are there, and we should take place. So now is that so what is the difference between a cell-based and NAPARS? So we'll just give you certain things that are required in the NAPARS and what is the major difference between the commercial protein arrays. So for example, if you say about the cloning, so in the cloning, NAPARS tagging is required, whereas in the commercial protein assays tagging required or not required. It depends on that. So printing molecules. So what are you printing on NAPA arrays? So if you just see, so what you are printing on the NAPA arrays are the cDNA, and in the commercial protein arrays you are printing the proteins. So here you need to print the cDNA. So if you just show you the overflow works again. So you have the cDNA template, and then the histag proteins just translating newly synthesized protein and is getting attached. Okay, so printing molecule cDNA. So manufacturing is that the different area. Expression system is cell free. So NAPA and the cell is the cell free expression systems, and the thing is that the cell based, which will have the bacteria, yeast, virus, etc. So amount of yielded amount of target proteins will have low yield, that is in micrograms, whereas others will have high yield, that is milligrams grams. Protein display in situ, that is just in time. So these are some of the important things to consider, which I'll just mark, like the printing molecules, the cloning, expression systems. So these are some of the important things that we should keep in mind when we are talking about cell-free assays and the cell-based assays. So then in laborious protein purification and acquisition, so which is one of the major challenges. Then affordable buying the proteins, long lasting, low economically cloning cost, high expensive plus purification plus price of the printed proteins. So you can see the cost. Applications. So now if we see what are the different applications. Detection of the chemical protein acetylation for human proteins. So these are all part of the publications that have been using different NAPA arrays and have been using find out different functions and the different structures and different applications of the protein microarrays. So, for example, detection of the chemical protein acetylation, which is your NAPA. So, in this, it has been reported that NAPA screen for human proteins or the 6,000 genes that undergo chemical acetylation. So that undergo chemical acetylation. So this is one of the PTMs or the post-translation modifications that can also be studied using NAPA. So these studies add chemical acetylation to the toolbox of the modified proteins. One or two general methods, that is single or multi-step uh, methods. So you can study the PTMs like acetylation using NAPA arrays. Similarly, to have to discover the protein-protein interaction. So if you have all the proteins, now you are giving the lysates over it. So what is the proteins, which particular proteins are getting interacted with it. So a protocol that is being presented to enable the study of PPIs at the protein level. So offers a third high throughput platform to interrogate the interaction. So it will also gives us an idea to have the interrogate the interactions. So interactions between your query and the immobilized target proteins are detected through addition of a fluorophore labeled halotag ligand. So between the query protein and immobilized target proteins. So what are the target proteins? So these are in a cell-free expression array systems when through NAPA system is getting transcribed and translated under mammalian milieu or the mammalian condition. So what it is happening is that it is you have the antibodies printed, and once the antibodies are printed you have those particular proteins which are getting stick to that particular antibody, and once this protein is getting attached to the antibodies, so then what it is happening is that it is getting immobilized. So the immobilized and the target proteins, what is that interaction pattern, that can also be studied through NAPA. Then profiling the interactome networks. So about the protein array, NAPA protein array procedures, then multiplex reproducible arrays, 5 different DNA plasmids, which significantly increases the array density. So the major thing for an ideal microarray should be to have high density arrays. So identified hits in protein interaction and your serum screening studies and number of display proteins per microarrays by 5-fold. And what are the biomedical research like the serum immune profiling with pre-invasive cervical lesions and invasive cervical cancer. So as I mentioned that it can be and a biomarker can be used for different diagnosis and the prognosis. So you can see so two risk contain high risk HPV types valuable for measuring the serologic response to HPV in cervical disease. Okay. Then diagnostic performance of individual tumor associated autoantibodies and autoantibody panels in ovarian cancer. That thing that we had shown for different brain tumors can also be used against in different cancers like ovarian cancers and other cancers. So again, we have started with an example of the autoantibody of profile characterization like the osteoarthritis and the rheumatoid arthritis, which shows that different autoimmune disorders and can be used for a uh, particular things. Then we have the host pathogen interactions like the human uh, pneumophila. So what it is there is that when you have the high density NAPA human arrays, human halolysate based cell-free expression system, so full length human open reading frames, binding parameters, then the targets identified, high fidelity of NAPA during physical protein-protein interaction screening, and the great potential for the interaction of the novel host. So these are all some of the applications that is being greatly used. So if I just give you about the case study of finding your particular so if I say your case study of the NAPA. So it just is that acetylation of what we have just spoken about is a post-translation modification and can be used. So these mediates many potent functions and the diverse biological process. So this is just an overview and abstract. So here you can see that how it has used. So it has used by the NAPA 2. So you can definitely go to that this manuscript and can go through it in much detail. So I'll just give you an overview of using of this NAPA technologies, that is to have to screen for different chemically acetylated and different other proteins. So you can see that a small set of firstly the positive and negative control genes, and it was printed on the test array. So for example, these are the genes that were printed and then were acetylate which had been shown to some degree of photoacetylation in vitro. So this enzyme was used. So once the proteins have been, so what is the concept? Is the concept is that once the genes have been expressed the particular proteins and have been immobilized on within the antibodies, then what you are doing with an acetyl transferase enzyme, so which has some for the other uh, expression of the autoacetylation, what it will do, it will definitely to some extent it will do the acetylation, and which will help you to which will help us to understand that whether we can detect that acetylation or not. And P53 are and histone 84 are the negative control genes, where proteins were substrate demonstrated substrates of acetyl transferase were cloned into a flag tag in vitro translation. Plasmids were printed on the array slide to immobilize. So, for example, alpha flag antibody each translated protein onto the array. So, we need to have that certain immobilized tag to immobilize those things onto the array. Otherwise, so which particular antibody it will be getting into was initiated HeLa cell based in vitro translation mixture. So again, the HeLa cell based translation gives you the mammalian milieu or the mammalian condition, mammalian milieu or mammalian condition, and was supplemented with a T7 polymerase to transcribe the which were under the control of a T7 promoter. And immobilized proteins, the quality of the assessed using a picogreen staining, and the protein was using a second alpha flag antibody. So it was tagged with flag protein. So all the anti-flag and the antibodies, it will be get immobilized. So now these were produced with approximately 6,000 genes. The plasmid DNA for this is from the DNSU collection center. So this, as we mentioned, that Professor Lever had huge made this collection where and encoded a C-terminal flag or GST tag, and in vitro the genes were splitted, resulting in about 1,500 genes per array slide. GST tag, flag tag, and the GST tag, the arrays were processed with the radio labeled and the protein expression. The some were produced with a translation mixture lacking the T7 polymerase, and one was with that. So what was the thing that finally, with the help of NAPA, you can see that the top candidate sample film of conformation of the full lacking T7 polymerase and the single steps methods. So sample of corresponding between the show notable increases between the control and the so if there is a T7 polymerase, there is a increases in the intensity. So it has been found out. So these are some of the examples which I just wanted to highlight you. You can obviously go to the paper and manuscript and can try to find out. So the basic concept here is to have plasmids or the cDNA extracts which codes for a particular gene with giving you the cell extracts and the mammalian conditions, which will give you the expression of that particular protein, like the T7 polymerase and all those, and then which you will have with tags, certain tags of an antibody, and your that particular proteins is getting immobilized to that particular antibodies. So this will give you a consistent proteins, and it will get tagged to it. So if your protein is tagged with GST, so it will get to the anti-GST antibody. If it's getting tagged with the flag, and it will get tagged with the anti-flag antibody. So this will give us and ensure that the proteins are getting immobilized, and then we can look for. Now, which we know that for the cell-based expression systems, the cloning are one of those most important steps. So among which you have the VPLR reaction of the gateway cloning steps. For example, you have the gene of interest, you have the donor vector. So with the BP reaction, you are going into the donor vector, getting the gene of interest, and from the donor vector, you are getting into the plus with the destination vector with the large fluorescence enzyme. You got adding it in the expression vector and then expressing it, and obviously you will have some bacterial antibacterial gene, which definitely helps you to see that the selected bacteria grows with those resistant to that particular antibodies. So now, as I mentioned, now we have gone to the biomarkers, but biomarkers are not something which we can just say on the basis of certain characteristics, though which we have looked upon and which we have identified, but still there remains different questions in context to biomarker identification challenges. So as I mentioned, that whether it is a population specific marker only, or it is a global marker which is expressing across the cohorts, and which is present, which should be present across those particular things. Then it is a sample specific marker, or it is just onto a specific set of sample preparation that gets the markers done, or it's just a control cohort, or it is a just uh, getting tagged with the sample cohort. So, and also the validation, like it is an independent validation or a blended validation on a different cohort that gives certain proteins or certain genes to be the associated with the biomarkers. So now, as I have mentioned, so there's some concepts of specificity and the sensitivity. So these are a classical example of a chart, that is what is called a confusion matrix, where you can see that there's true positive. So if you have, so your marker, so if you just think in a way, you have selected one biomarker, and your biomarker is able to classify the disease state into disease, and normal condition into normal. So that is the true positive and the true negative. So who are diseased, it is true positive. Who are not diseased and not diseased are true negative. But if it is happening that the person is not diseased but still giving, so that is known as your type 1 error. The predicted value is positive, but it is false. And the type 2 error is the predictive error is negative, but it is positive. So this again, you have to understand that at which condition which one is very much detrimental for you. So if you are having a certain set of biomarkers that you are trying to say that it is able to uh, predict whether it is in a disease condition or not, or certain particular uh, you have a particular set of machine or instrument, medical instruments, to say that it is a particular disease condition or not. So when you are saying that a person is diseased, but still you are uh, saying that yes, it is be it negative, no worries, that it is that a person is in a disease condition, but you are saying that it is not disease. That is the false negative. It is not. It is not negative. It is actually positive, but it is saying negative. So then that kind of biomarkers would become very much. Then that will become very much detrimental, because a diseased person would be treated as a non-diseased one. So this is a basic score. So that what is one score, that is a recall. So false positive rate or the true positive rate is the false positive divided by the true negatives and the false positive that we are getting, and that positive rate is the true negative by true negative plus false negative. And so this is the accuracy score, that is true positive plus true negative divided by all. And so this is the sensitivity and the specificity difference. Uh, so which is very much important in as a measure for when we were doing analyzing of this particular biomarker. So now with this condition of kind of specificity and accuracy and different number of samples, you can calculate the specificity and the sensitivity from the given formula and try to find out. Okay. Yeah, so this is all for the this week with various technologies, various characteristics of biomarkers, when we can classify the biomarker, what is a biomarker or what is an ideal biomarker, with the technology of NAPPA, uh, which has helped us in overcoming the challenges for high density protein arrays, which is associated with protein purification and the expression, be it in the abundance based or the function based arrays, where we can do in an in vitro system, in vitro translation system, translation mixture, which we have the plasmids or the cDNA plasmids, and which we can use different mammalian system to have the mammalian condition and to have a natural folding, which will give us the protein and tagged with certain antibodies to get immobilized, and then we have the query proteins to get have an interaction and can be and has been used across with various techniques which we have seen across with the various techniques with various applications of NAPPA. Yeah. So if you don't have any question, I think then we can uh, end today's class. Yeah. So thank you all. Yeah.